Welcome all to this session. This is Thelma, Assistant Professor, Department of Zoology, Patimo College. In this video, you will be learning about the physical methods of sterilization. Sterilization involves the destruction or removal of all living microorganisms, spores, and viruses on an object or in an area. The agents that kill microbes are called as microbicidal agents. If the agent specifically kills bacteria, it is called as a bactericidal agent, and if it kills a fungi, it is called as a fungicidal agent. There are many physical and chemical methods that are capable of destroying the microorganisms on non-living materials or even the skin surface. However, once these materials, when exposed to the air and surroundings, there are chances for them to get contaminated again. Heat is one of the most important physical methods that is fast, reliable, and relatively inexpensive. Above the growth range temperature for a microbe, the enzymes, proteins, as well as the nucleic acids are denatured. Heat also removes water out of the cell, and this removal may also be fatal to the organism. The killing rate of heat may be expressed as a function of time and temperature. For example, the bacilli Mycobacterium tuberculosis are destroyed in 30 minutes at 58 degrees Celsius, but in only 2 minutes at 65 degrees Celsius and in a few seconds at 72 degrees Celsius. Each microbial species has a thermal death time. It is the time necessary for killing the population at a given temperature. Each species also has a thermal death point, which is the minimal temperature at which it dies in a given time. The next method of physical sterilization is incineration. It is using a direct flame to kill the microbes rapidly. For example, the flame of Bunsen burner is employed for a few seconds to sterilize the bacteriological loop before removing a sample from the culture tube. Flaming the lip of the tube also destroys organisms that happen to contact the lip while burning away lint and dust. Other examples are disposable hospital gowns, the bodies of the deceased victims, and other certain plastic apparatus are also sterilized using this method. The next method of physical sterilization is the use of dry heat. The best example that could be given for dry heat is the hot air oven that uses radiating dry heat for sterilization. This type of energy does not penetrate materials easily, and therefore long periods of exposure to high temperature are necessary. For example, at a temperature of 160 degrees Celsius, a period of 2 hours is required for the destruction of bacterial spores. Higher temperatures are not recommended because the wrapping paper used for equipment tends to char at 180 degrees Celsius. The hotter method is useful for sterilizing dry powders and water-free oily substances, as well as for many glassware such as pipettes, flasks, and syringes. One more advantage of this dry heat method is that it does not corrode sharp instruments as well as glass surfaces. This picture shows the use of direct flame as sterilizing agents. In this picture, the inoculation loop and the culture tubes are directly shown over the flame to remove any unwanted microbes. The next method is the use of moist heat. Boiling water is an example of moist heat that penetrates material much more rapidly than dry heat because water molecules conduct heat better than air. Therefore, moist heat can be used at a lower temperature and shorter exposure time than for dry heat. Moist heat kills microorganisms by denaturing their proteins. Denaturation is a condition where the chemical or physical property of a protein is completely lost. Moist heat can also be used in the form of pressurized steam, and this is regarded as one of the most dependable methods for sterilization because even bacterial spores are killed in this method. When the pressure of the gas increases, the temperature of the gas also increases proportionally because steam is a gas. Increasing its pressure in a closed system increases its temperature. As the water molecules in the steam become more energized, their penetration increases substantially. Examples for this method are autoclave and pressure cooker. Sterilization by moist heat can also be done without pressurized steam. Liquids and other objects can be sterilized by exposure to free-flowing steam at 100 degrees Celsius for 30 minutes on each of 3 successive days, with incubation periods at room temperature between the steaming. This method is also called as fractional sterilization because a fraction of sterilization is accomplished on each day. This method is also called as Tyndallization because it is named after its developer John Tyndall. During the first day's exposure, the steam kills all the microorganisms except the bacterial spores, and the object is incubated overnight. During this incubation period, the spores germinate and the viable cells multiply, and these viable cells will be killed on the second day when they are exposed to 100 degrees Celsius. And again the material is cooled and any remaining spores are allowed to germinate and the resulting cells only to be killed on the third day. The most important method which employs moist heat without pressurized steam is the pasteurization. Pasteurization is a method by which the bacterial population of liquid such as milk they are destroyed that may cause spoilage and human disease. An important note is that spores are not affected by pasteurization. One method for milk pasteurization is called as the holding method or also called as batch method, which involves heating at 63 degrees Celsius for 30 minutes. Two other methods are flash pasteurization method at 71.6 degrees Celsius for 15 seconds and the ultra high temperature method at 140 degrees Celsius for 3 seconds. The next important physical method of sterilization is the filtration. Filtration is a mechanical method that can be used to remove microorganisms from a solution or gas. The most important type of filter which is used in this technique is the membrane filters that consist of a pad of cellulose acetate or polycarbonate mounted in a holding device. As fluid passes through the filter, organisms are trapped in the pores of the filtering material. The solution dripping through the filter into the receiving container is decontaminated or it is said to be sterilized. Heat-sensitive liquids such as beverages, bacteriological media, toxoids, and many pharmaceutical and blood solutions are sterilized by this method. The previous method is a technique by which liquids are sterilized by the usage of filters, but air can also be filtered to remove microorganisms by the use of high efficiency particulate air filter, in short, which is called as HEPA filters, which consists of mat of randomly arranged fibers that trap particles, microorganisms and spores. As a part of biological safety cabinet, HEPA filters can trap over 99% of all particles, including microorganisms and spores, with a diameter larger than 0.3 micrometer. The air entering surgical units, hospital wards for respiratory diseases, and pharmaceutical filling rooms are sterilized using HEPA filters. In microbiology, all works are performed inside a laminar airflow shown in this picture. The air entering inside and leaving the laminar airflow is filter sterilized by the use of HEPA filters attached inside the chamber. The next method is the use of radiations to kill microorganisms. Ultraviolet rays and ionizing radiation such as X-rays and gamma rays have the ability to kill microbes naturally. UV radiations. Ultraviolet rays has a wavelength between 100 and 400 nanometer with the energy at about 265 nanometer, most destructive to bacterial cells. When microorganisms are subjected to UV light, cellular DNA absorbs the energy and adjacent thymine molecules link together, kinking the double helix and disrupting DNA replication. The damaged organism can no longer produce critical proteins or reproduce, and it quickly dies. Ultraviolet light cannot penetrate liquid or glass, and hence it is used to decontaminate hospital room, morgue, pharmacy, toilet facility, or other food service operations. Ionizing radiations, such as X-rays and gamma rays, have wavelengths shorter than the wavelength of UV light. As X-rays and gamma rays pass through microbial molecules, they force electrons out of their shell, thereby creating ions. And this is the reason why they are called as ionizing radiations. The ions quickly combine mainly with the cellular water, and the free radicals generated affect cell metabolism and physiology. The heat-sensitive substances such as vitamins, hormones, antibiotics, as well as certain plastic and suture materials are sterilized using this method. The next important method is the use of osmotic pressure. Preservation by salting is based upon the principle of osmotic pressure. When food is salted, water diffuses out of the microorganisms towards a higher concentration of salt and lower concentration of water in the surrounding environment. This flow of water is called as osmosis, which leaves the microorganism dehydrated, and finally it leads to the death of the organism. The same phenomenon it occurs in highly sugared foods such as syrups, jams and jellies. However, fungal contamination and growth at the surface may occur because they can tolerate low water and high sugar concentrations. The last method of physical sterilization is the use of low temperature. Low temperatures found in the refrigerator and freezer retard the spoilage by lowering the metabolic rate of microorganisms and thereby reducing their rate of growth. Spoilage is not totally eliminated in cold foods. However, psychrotrophs remain alive even at freezing temperatures. This table shows the summary of physical agents used to control microorganisms, which involves incineration, hot air, boiling water, pressurized steam, fractional sterilization, pasteurization, filtration, ultraviolet rays, gamma rays, X-rays, dehydration, refrigeration or freezing. This flowchart shows the overall picture of physical methods of control, which includes heat, filtration, radiant energy, and low temperature. Under heat, the dry heat and moist heat are the methods by which bacteria and other microorganisms are killed. The dry heat employs hot air oven and incineration to kill microorganisms. When moist heat is considered, it includes boiling water, steam, and pasteurization. Under filtration, it employs the usage of membrane filters and HEPA filters. Membrane filters are used to sterilize liquids, whereas HEPA filters are used to sterilize air. The next category is the radiant energy that includes UV light and ionizing radiations. Ionizing radiations includes X-rays and gamma rays, which leads to the death of the organisms. The last category is the low temperature in the form of refrigeration and freezing. Hope you all enjoy this video. Thanks for watching.